All right, so today's microseminar is Sarah Lebeis. So Sarah did her undergrad at Michigan State, and then for grad school went to Emory and worked in Dan Kalman's lab in pathology. So she was actually doing mouse stuff for her grad school, and then I first met her when she interviewed for a postdoc in Jeff Dangle's lab, where I was a postdoc at the time. Every time Sarah and I introduce each other, we like to tell our origin story. So basically, the short version of this is when I interviewed for my postdoc in Dangle's lab, I was deciding between Rich Lenski's lab and Dangle's lab. Dangle sat me down and said, you know, you can do great things in Michigan State and in Rich's lab, but you've been doing those things before, or you can come to North Carolina and do something completely new and jump into the complete unknown and do something great in a completely unexpected way. And so that was basically challenging my adventurous spirit, and so that was basically what won me to go to North Carolina. So I thought I was alone in this, but apparently Dangle uses that line on everybody, because she got the exact same speech, and we only found out when I was leaving the lab, when we were toasting for, you know, when I was my last postdoc party there, and we were toasting, and I told the story, and then Sarah realized that we had both been conned by the same line from Dangle. All right, so in 2014, so after postdocing in Jeff Dangle's lab, where she did a variety of awesome things, some of which she'll probably touch on today, Sarah started her own lab at the University of Tennessee, Knoxville in February 2014. And I will leave it at that. So today's seminar is Sarah Lebeis from Tennessee, Knoxville. If you're on here on Google Hangouts, please mute and turn off your camera. And Sarah, please take it away.
Thank you. I'd like to start by figuring out how to share my screen, probably. And I'd like to thank Dave for inviting me and thinking of me. Thank you for the introduction. I'm happy for you to give it, because that's a true story, and it's an effective line, which I think for all professors out there looking for recruit, I think that's a great line that you should use to recruit students to your lab. I'd also like to thank Jennifer for contacting me in the first place to do this. I'm excited to try a new venue and see how this type of talk goes. I'd also like to thank her. When we were both at UNC, she introduced me to a product called MoBio Power Soil Kit. And I'm sure MoBio will also thank her, because I have spent a lot of money on that kit, and hopefully you'll see that the research that came out of that has gone a little bit of a ways. So the work that I'll tell you about today, as Dave said, is stuff that started while I was a postdoc in Jeff Dangle's lab at UNC, and then it's stuff that we've continued here at my own lab. So there'll be some more stories that are going to be sort of an extension of that, as well as some new directions that we're going in. So I'm probably going to breeze through this pretty fast, so there's plenty of time for questions. Please feel free to write them down and ask me at the end, because I'm sure there will be time, and I'm happy to talk about it. So basically, the outline is that I'm going to tell you a little bit about my history and my story about how I made this leap into plant microbiome, especially root microbiome, and then I'll tell you about old work, new work, and sort of continuing work from there. So the way that I like to think about it, so as Dave mentioned, I did my graduate work in mammalian pathology, and I think one of the things that I didn't appreciate at the time, and we really couldn't appreciate at the time because we couldn't really get a handle on it, is that all of our experiments about the molecular mechanisms for how diseases work and how the immune system works does not exist within a vacuum. We live in a microbial world. Everything that we see, taste, touch, and most definitely smell is coated with microbes, whole microbial ecosystems. And so as I was finishing up my PhD, this really sort of became a question we could start to answer, because sequencing technologies sort of revolutionized the way that we could ask this question of who is there, how can we characterize our microbial world. And so I think that the soil is a great place for this. I get really excited about this because if you think about the system that I'm working with, the roots, the inoculum for these roots are coming from one of the most microbially diverse environments on the planet. So what's really fascinating to me is how can you go from something that is so diverse to something that is remarkably consistent and robust. over time and between different genotypes. And so what I'm going to talk about today are some of the processes that I think are going on, both on the plant side, so how does the plant control which microbes can enter the tissue, the root tissue, and also on the microbe side. So I'll show you certain microbes that we think colonize the roots really well, and then sort of downstream from that, how do they either prevent or promote subsequent colonization of this really microbially diverse environment. So this is one of my standard disclaimer slides. All of the slides you are about to see are based in Arabidopsis thaliana. It has its limitations, certainly. The fungal communities within these will be different because they don't have the characterized mycorrhizal interactions. And so that sort of brings me to my second part of my disclaimer. The stories you're about to see are also bacterial in nature. And that's not to say that fungi and other eukaryotes don't play a really important role to the overall health of the plant. And on the opposite side of that, the viruses don't also contribute. But this was sort of my starting place for this. And the reason for that is because we wanted a system where we could characterize things really well, and there are amazing databases for bacterial 16S. And we wanted a plant that had the genetic tools available to it, like Arabidopsis, so we could hone in on really particular mechanisms that might be driving the bacterial communities that we were looking at. And so when we think about plant-microbe interactions, of course, all of this occurs along a spectrum. So depending on how the microbe affects the plant health. So the plant is the central perspective that I'll be talking about. And so this can be negative or pathogenic. It can be positive or mutualistic. But that's not to say that the way I think about it is as a spectrum. It's not three separate categories, and it's not set in stone. So something that is commensal in one set of abiotic conditions may be mutualistic in another. And so when we think about this, this is a moving spectrum. It's not just three distinct categories. So on the pathogenic side, again, there are a diversity of microbes that cause these phenotypes. So you can also have viruses or oomycetes or fungi causing pathogenic phenotypes, and then obviously bacteria is what I'll focus on today. On the mutualistic side, that's also true. So so here is a bacteria on the top, a rhizobium that's forming nodules. This is considered a mutualistic relationship with the plant. You can also have the mycorrhizal fungi at the bottom here, which are helping to bring in nitrogen and phosphorus for the plant. And so when we think about the different categories where things can be mutualistic, the three main things that people tend to think about are nutrient absorption by the plant, so microbes helping plants to get nutrients. They can help with growth. I'll go over some examples of this in the next slide. And then you can also have protection that gets induced by these mutualistic bacteria against the pathogens over here on the left. So those are the sort of the three big categories that we think of. So getting into some of the more, let's go back a little. So thinking about some of the more, I just changed the batteries on my mouse and I think it got a little too eager with clicking. So thinking about the different categories of things, so the benefits that the microbes can provide with the plant, of course you can have that they help to provide nutrients. They can actually produce growth-promoting hormones such as auxin. They can provide protection against soil microbes either directly or indirectly, so they can just sort of fill the niche and take critical nutrients away so the pathogen can't come in, or they can induce what's called either induced systemic resistance, or in the leaves you also have systemic acquired resistance, which can help prevent subsequent pathogenic infection. On the other side, obviously the main thing that the plants provide the microbes is sort of food and a nice, safe, warm place to live. And so within the exudates that come out of the roots, you can have things like organic acids, amino acids, various metabolites, and sugar. So it's estimated that 5 to 20% of the carbon that plants will fix is actually secreted through their roots and becomes a really yummy carbon source for the microbes. So the microbes, again, that I'll be focusing on today are in the root. So we call the above ground tissues the phyllosphere. So I won't be talking about phyllosphere microbiome today. Instead, I'll be talking about root microbiome. And so for this, there are three types of samples that I'm going to focus on. I'm going to focus on the bulk soil. So that is the soil when no plant is present. The rhizosphere. So the rhizosphere is the soil that actually coats the root system. I won't be talking about rhizoplane, which is actually sort of the microbes that strongly attach and sort of form a biofilm on the surface of the root. But I will be talking about the endophytic compartment sample, so that's microbes that are actually inside of this tissue. I'd like to give an initial shout out to this gentleman down here on the left, Derek Lundberg. Not only is he a terrific artist drawing this sketch right here of an Arabidopsis thaliana plant, but he is the gentleman that I ran into in the interview that David was talking about at the beginning, who told me about this project. And I thought, what an awesome project to get involved with and see how the immune system might help to control the root microbiome membership, so which microbes can actually exist within here. And so this is from Derek's original paper, which was really to help us characterize what's there. So if we separate the microbial communities into these three fractions, what does it look like? So we did 16S sequencing on the V8 region using, I think we used 454 sequencing at the time. And we generated a bunch of operational taxonomic units, and we asked who is there. So here they've been assigned by phyla. And so what you can see is that soil and rhizosphere look really similar to each other, both in phyla distribution as well as in the overall diversity of what's there. Once you move inside of the root, this is sort of the phenomenon I'm talking about. You have a decrease in diversity, and that is characterized down here by the phyla distribution, where you see a massive expansion of things like Actinobacteria, and then we see changing abundances of several proteobacteria. So here the proteobacteria look like they go down, but if you actually look at different classes, some classes go up and some go down. There's a really dramatic shift in what the communities look like outside the root compared to inside of the root. And so the first question we had with this is there a core microbiome? So within the Arabidopsis thaliana roots, can we see consistency in who is there? How robust is this phenotype that we want to look at? And so for this, there's a set of parallel studies done in our lab at UNC in North Carolina. And then we partnered with a group in Germany, Paul Schutze-Leffert's group at Max Planck in Cologne. And using Arabidopsis in both cases from soils from our sort of designated areas, we asked, what does the microbiome look like? What things are conserved and what things are different? So what things are dependent on whether a soil comes from here or comes from the US or comes from Germany, and what things are just this process of going from soil to root. And so here is some of our data from that, which you can see is we took, in this case, there were 8 different ecotypes, so we also layered genotype on top of that. I can get into that if you guys have questions later, but I'll also focus on a genotype component later in my stuff. So we took 8 different genotypes and we grew them in 2 different soils. So CL means Clayton, that's from Clayton, North Carolina. MF means Mason Farm. Mason Farm is actually right next to UNC's campus, so it's incredibly handy to get to. We had 2 different time points. We have young and old, and then we have the 3 sample fractions that I've been talking about. Soil, so this is just bulk soil. We have rhizosphere, so the soil outside the root, and EC is are the samples inside the root. So here, this is a principal component analysis of weighted unifrac distances. So basically, spots that are close together have very similar communities. Their OTU abundances and everything look really similar. Things that are far apart are very different. So the first thing we see is that the #1 component of what your microbiome looks like, basically, is whether you're outside a root or inside of a root. So everything over here is soil and rhizosphere. Although the soil and rhizosphere do separate from each other. I need to stop using that mouse. And within the endophytic compartment, you can see that everything ends up on the right. The second determiner for what your microbiome looks like is which soil you came from. So all of our Mason Farm samples are at the top of the chart, and everything on the bottom is our soils, or is our Clayton soil sample. So basically, more than what microbiome you start off with, it's where you are that sort of determines what your microbiome will look like. So here we're looking at we're looking at a heat map of the abundances for all of the OTUs. All of the OTUs are at the top. So everything here at the top is a different OTU, and then the samples are denoted over here on the side. Again, you can see that the biggest break is whether you're outside of a root, so soil and rhizosphere are at the top, and endophytic compartment is at the bottom. So what's really cool is you can see a couple of different groups form. So everything in the box labeled 1 right here, these are things that are abundant in the soil, and then they seem to get excluded from the internal component. The focus of the rest of the talk will be on the immune system, and so this is sort of the microbes that I'm going to be looking for. These are things that are getting excluded, we presume, because of an immune system or some other selection by the host, or even at the end of the talk I'll talk about microbe interactions that are preventing these things that are highly prevalent in both soils from actually being able to get inside. And then we also have what we call EC sort of EC microbiomes. So these are core enriched things. So these are things that are not as abundant in the soil that get heavily enriched once you move inside. And these are the things that we sort of think are providing some sort of function, or they may have some sort of activity. This is a new line of research we've gotten into where we're looking at what is it about these microbes that make them really good at colonizing. And so that's something else I'll touch on today. So the experimental design for what we're talking about is we're starting with one common microbial environment. So for the first set of experiments I'll talk about, it'll be a wild soil. It'll be soil from Mason Farm, and then we'll plant different genotypes of plants in there. We'll ask what's the same. This is our core microbiome, and then we'll ask what's different. These are things that could potentially be driven by the genotype differences between the different plants. This is something we'll start off with in wild soil, and then later on I'll show you a synthetic community we built of 38 bacterial isolates, which we have the whole genomes of, so we can ask some really direct questions, which coming from pathogenesis background, I love having direct questions available for such a big question. And so the question will be, does the plant immune system control the root microbiome composition? And then also we'll be able to touch on what taxonomic level we think this selection might be occurring in. So here we have sort of an overview of the different types of immune mutants that I worked with. First of all, I did PR and NLR signaling components. So the idea with this mutant set is if you can't see your microbial environment, can you properly respond to it? And then part of that response was we looked at endogenous propeptide production and whether that impacts root microbiome. And then the last part is that we also looked at phytohormone communication. So also downstream of the PRR and NLR signaling, did we see that phytohormone communication caused differences in the root microbiome? And so I'll actually start off with the third set first. And these are the different genotypes that I used. So I had a quadruple mutant, which could not make jasmonic acid, could not make salicylic acid, and could not sense ethylene, as well as 2 singles within the salicylic acid part of that genotype. I also have a triple receptor mutant, which cannot sense any of these 3 phytohormones. And then on the other side of that, I have 3 salicylic acid mutants, which are constitutively making this phytohormone to ask when something's being made too much, does that also affect it? So here, this is another principal coordinate analysis. As we expected, we have really good separation between the external communities and the internal communities. So EC, again, is all over on the right side. But the next question was, we see quite a bit of spread in these EC communities. Is part of this difference driven by the genotype? So here is another phyla distribution. So again, soil and rhizosphere look really similar to each other, both in composition of their OTUs at the phyla level, as well as the diversity. And you see a difference in the EC. So when you move inside the EC, even in the wild type, you see that expansion of Actinobacteria we saw, and we see differences in the Proteobacteria as well, and a decrease in the diversity. So one of the things which I got really excited about is that we have differences within each mutant set. So these are the guys that make too much salicylic acid. These are the guys that can't make salicylic acid. These are the guys that can't sense salicylic acid. Within each of these sets, we have significant differences way out at the phyla level. For us, this was really exciting because before this, the only genotypic differences we had seen were zoomed way down to the OTU level. So way out at the phyla level, we're seeing differences. So it looks like genotype is contributing in a large part to what the microbiome looks like. The other thing is that we have differences in different phyla. So Actinobacteria are affected, Proteobacteria are affected, Firmicutes are affected. There's sort of big differences we're seeing in lots of different things. And so zooming in on the EC in particular, since we see such a difference with the genotypes, we wanted to start looking at what families might be contributing to this. Is it a single family that's driving this, or is it sort of all of them that are driving these phenotypes? And so the phenotype I'll focus on first is the Actinobacteria with CPR5 and PAD4. So here, Again, we had a significant decrease in Actinobacteria for our SA overproducer and PAD4, which doesn't sense salicylic acid. And when I looked at the family level of this, there were no significant differences compared to the wild type for CPR5. I can go into that later, what we think is going on with CPR5. But with PAD4, it did appear to be driven by a single decrease, so that the Streptomyces within the Actinobacteria were significantly decreased. And this decrease that we saw over here is something that we did not only sequencing on, but we also did a more quantitative assay. And I actually did bacterial counts using cardfish, so a type of fish that's amplifying signal, and I spent a lot of time looking at microscope slides. So if you want to talk about it, I'm happy to talk about it because I've spent a significant portion of my life counting bacteria. So we did not see any of these differences with the other genotypes, but we wanted to know this is something we can eyeball, right? That's not a very systematic way to go through what might be contributing to these differences. And so for the next set of studies, I enlisted the help of this gentleman down here, Sur Herrera Paredes, who is still in Jeff's lab, and he is still my statistical guru. He has saved more than one life, including mine, trying to make sense of these giant phenotypes we see and try to understand what's going on. And so we used a zero inflated negative binomial in this case to look for the differences between each of the mutant classes and wild type. So over here is each dot is a significant either enrichment if it's above the red line, or depletion if it's below the red line of the relative abundances for, so each dot represents a family, where Colombia is represented by the red line. So what you can see is that different mutants have different number of differentiating families. So CPR5 and the quadruple mutant depths, for example, well, and PAD4 and SID2 have a lot of differential families, and also the distribution of them is very different. So if you compare CPR5, you have about equal number of enrichments and depletions, whereas with the quadruple hormone mutant, which isn't making jasmonic acid, salicylic acid, and can't sense ethylene, you have mostly depletions in this case. So what is the taxonomy of these things? They're just black dots right here. So if we, so this is unweighted distribution, so we just took all those dots and we assigned them to which phyla they belong to. So one of the cool things that's come out of this is we can see, for example, we had a significant, for CPR5, we had a significant decrease in Actinobacteria. Well, this model has picked out about 9 different families of Actinobacteria that are leading to this decrease that we saw way out at the phyla level. The other thing that I got really jazzed about is we asked, can we use this phenotype to be able to group different genotypes together? So if our phenotype is enrichment or depletion of families, can we use that to be able to link it back to our genetics? And so in this case, what we've done is we've taken this enrichment and depletion profile, and we've calculated the Manhattan distances, and then we followed that up with Monte Carlo testing. And so what we can see is that the quadruple mutant and its 2 single mutants actually group together really nicely. So these enrichments and depletions seem to actually be driven by, or potentially driven by the genotype. And this is something that we obviously went back and looked at, but this was just sort of a marker. The thing that they all have in common is altered salicylic acid, so that sort of helped us zoom in on that. And not only is it just that they belong to the same phyla, but they're actually the same family. So if you look over here on the depletions, there were loads of depletions in the quadruple mutant, but half of them are shared with SID2. So half of the depletions that we see are appear to be caused by just the lack of salicylic acid production. We were asked, does this hold true? So far we've seen that families are driving phenotype, or families are driving phyla, are OTUs also driving this? And so here, this is looking at all of the OTUs that are within a certain family. So on the left, we have a particular Actinobacteria family. And so you can see that what we expect to see from this is an enrichment in CPR5 and a depletion in depths in SID2. Well, what you can see below for these OTUs that belong to this family is that it's not a single one that's driving it, it's multiple ones, and you have this sort of overlapping pattern. So it seems that it's not a particular OTU that's driving this, but rather at the family level that we think this is happening. And a couple of slides, I'll get into the fact that we don't think it's just OTU. So this isn't just a matter of sequencing. When we try to repeat these experiments with individual bacterial isolate strains, we see something really similar. So that's really cool. So from this, we think that salicylic acid is modulating the composition of these of the root microbiome. and that we see differences at phyla, family, and OTU level, but it's not a single OTU that's driving these differences. The selection or the change appears to be happening at a higher level. And so one of the first things we did is we constructed what we call a synthetic community or a reconstructed community. So here is an example of the one that we used. So there were 4 different phyla that were represented. There were 38 isolates and several families. So one of the cool things that I just mentioned is because we have multiple isolates within the same family, we can start to ask whether this selection that we think we're seeing, is it occurring at the family level or at the individual strain level? So we can just skip over that whole OTU thing, which is kind of fun. Okay, so here what we've done is we've taken those different genotypes, so they're all nested within here, and we've given them these microbes at the bottom. So they're colored by phyla here. What you can see is there's 2 main sections. There are those things that robustly colonize, so these are things that are present in the wild type samples more than 50% of the time. So these are things that are consistently getting in, and you can sort of appreciate that from the zoomed out level by seeing that the abundance is generally higher in this box than it is this box. This box is darker than this box. There's more white over here. These differences in abundances pushed the clustering over here. So this was unsupervised, and what you can see is these abundances are leading to differences between the endophytic compartment, the bulk soil, and then our initial inoculum is down here at the bottom. So that was kind of cool. Another thing which I'm going to touch on, which I got really excited about, is if we ask, so there are different genotypes in here, we're going to ask which of these isolates are different in the genotypes. So I've highlighted them here. You'll see that the majority of them are over here on the sporadic or non-colonizers. So it's not the things that robustly colonize a plant, it's those things that we expect to be sort of excluded. And one of the cool things is that if you look at the abundances, in the mutants they're higher. So things that should be getting excluded from a wild-type plant are now present at higher abundance when you have a mutation. So that's really cool. And so this is pulling them out in their own separate sort of heat map. So this is their wild-type abundance. And then when you have the different mutants, you have red box means enriched. You see mostly red boxes over here. These are things, again, that were depleted, when you compare them to wild-type. Another cool thing, which unfortunately was not statistically significant, but I can appreciate from a zoomed out level, is you see that things that tend to be enriched in PAD4 also tend to be enriched in the SID2 mutants. So this was still pointing to the idea that salicylic acid might be one of the drivers. So one of the follow-up things we did is we took plants and we just sprayed them with salicylic acid to say, if we give them salicylic acid, will that change this community alone? And so I'm going to show you 3 examples where the answer was yes. So over here on the left, what we have is an isolate that, this is its abundance in the inoculum. You can see that it has no difference between soil and soil sprayed with SA. However, if you look in the endophytic compartment, there was a statistically significant difference in the ECs of plants that did not or did receive salicylic acid. And we were able to follow that up with growth curves. So here in buffered media, you can see that if you have 370 with increasing amounts of salicylic acid, you have decreasing growth, which was really cool. The opposite phenotype is also true. So here is another isolate. This is 273. So with 273, when we add salicylic acid, both in the soil and the endophytic compartment, we actually have better growth. So this is really cool. And then over here on the growth curve, again, when you have increasing amounts of salicylic acid, you have better and better growth of this particular isolate. 303 is my favorite isolate from the synthetic community. I can talk about that later if you're interested. But basically what happens is, again, we have an increase in salicylic acid, an increase in relative abundance when salicylic acid is present. And within the genome of this, we actually had matches for every enzyme in a salicylic acid degradation pathway that had previously been characterized in Streptomyces. So this is a strep. Also, when we grow this isolate on minimal media where salicylic acid is the sole carbon source, it does grow. So that indicates to us that that degradation machinery may indeed be functional, although I think in the real world it's probably not using salicylic acid as a carbon source. There's probably, it's probably being co-opted as another molecule for another excellent thing to help this thing grow. So I've told you about the reconstruction community that we did, that we used to show that things that we isolates that we get from EC actually can colonize EC. That's always nice. We can see that sporadic and non-colonizers tend to be the things that are enriched when you're missing part of your immune system, and that salicylic acid alone can change community composition. So what I'll go into next is sort of a quick study of upstream of salicylic acid, as well as we're starting to move away from just who's there to what are they doing. So again, with this same setup, we're going to look at how things that are upstream of salicylic acid might control the composition, and then how does this actually relate to function in these different communities. So the group that I'll focus on today are the PRR and NLR signaling components. We also checked the peptide signaling mutants, and we did not see very many significant differences, although we didn't go back and look at function again. We still need to do that. So here, these are the mutants that we use. So we're using either co-receptors or PRRs. So these are things that shouldn't be able to see the microbe-associated molecular patterns, and then also the NLR signaling components. Again, we see that soil and rhizosphere on one side, endophytic compartment is on the other. This is something that I think is super cool, and I wish this wasn't covered up, but whatever. So here, this is a pattern we always see, right? So if we're looking at taxonomy, so this is just the 16S over here in terms of diversity. So here, soil is diverse, rhizosphere is about equally as diverse, there's not really a big difference. We always see a big drop-off in the EC. However, what we did is we took these 16S sequences and we used PyCrust to infer function. Now, we didn't just use a standard database. We created our own database that was limited to plant or soil-associated microbes to try to get a more accurate handle on what these things might actually be doing. But this was a trend that we're sort of interested to follow up on. Basically, if this is the number of functions or activities you expect to see in this community here, it actually goes up in the rhizosphere, and it does not drop off when you go to the EC. I sort of like this because I think it points toward a hypothesis that is there aren't as many just sort of hangers-on. If you're getting to the EC, it's for a reason. It's either because you have an activity or because you're performing a function. And I think that's one of the exciting things that we can sort of delve into. So here, if we use that PyCrust data, and we're using the K-graphology or the KO numbers, and we're asking what is driving it in different directions. So if soil and rhizosphere are over here, endophytic compartment is over here, what are the different factors that are driving it in different directions? One of the interesting things, which you'll see later too, is that one of the major drivers for whether you're on the left with the EC or the right is whether or not you have a bunch of ABC transporters in you. And so we're sort of excited to look at what the potential things are that they might be transporting. Another thing that's really cool, down here we have soil at the top and rhizosphere at the bottom. These KOs down here are things that are associated with a two-component system that's important for chemotaxis. So whether you're just hanging out in the soil by yourself or actually moving toward the surface of a plant, or whether or not you have the ability to chemotax, which I think is really cool. So here, this is just briefly to go over taxonomy. Basically, again, we see enrichments and depletions, but what I really want to skip forward to show you is that the enrichments and depletions and how they relate to the functions on the left, I think, are sort of an interesting story. So if we start with the first mutant here, BAC1, again, this is comparing it to Columbia. So you can see there was one family that was enriched and 14 that were depleted. However, when you look over here at those same samples, so Columbia, if these are the functions that we predict are in those samples, when you go to the BAC1 mutant, you can see there's a pretty big variation, but also a drop-off in those potential functions that are there. So these 14 things matter. However, when you move over here to the BAC1 BKK1 double, there were no enrichments and there were 35 depletions, but when you look back at the function, that's not the case. There's not a drop-off in function, and there's actually a pretty tight distribution of the functions that we infer may be there. The third thing, this is the last one I'll go through, I promise. If you go to the triple mutant, the BAC1, BKK1, CIRC1, there's only 5 families that are predicted to be depleted. However, you see the same drop-off and spread that we saw with the BAC1 mutant. So now what we can do is go back and look at the overlap between this triple mutant, the double mutant, and the single mutant to try to infer which families might be driving these differences that we infer over here on the functional side of things. So this is kind of an interesting direction that we're really excited to go into. And as you'll see, it's not just limited to sort of PyCrust data. We also have some isolate data that we're excited about. So this isolate data, again, comes from the fact that we have genome sequences from our synthetic community. So if we go back to that synthetic community that I told you about in a little while ago, the question is, what are the functions associated with these robust colonizers that are not present over on the sporadic side of things? So here, again, we're looking at KO pathways. We're looking at the incident rate difference. So basically, if a KO occurs on the robust side and does not occur on the sporadic side, the difference between them is how we generate this vector. And so, gosh, this mouse, I should not have recharged its batteries. It's got a life of its own. Okay, so again, as I mentioned from the pie crust data, a lot of the difference between whether you're what we think is going to be a robust colonizer or excluded from the microbiome is whether or not you have these ABC transporters. So one of the things that's super cool is that a lot of these things tend to be in the same thing. So these top 3 hits are all predicted to be within the same type of metal transporter. Everything in the middle is predicted to either be a transporter of carbon or amino acids, or sort of things that we sort of expect to see that are important for things that can actually get inside and sort of utilize the best of their environment and be able to grow faster and better. Another super cool thing we have in the enriched guys is the exact same KO that we saw from the pie crust data for this 2-component system that's important for chemotaxis. So the last thing, which I actually forgot to mention earlier, so I'm glad I have a slide here, is that if you'll remember, when we looked at the synthetic community, there were multiple isolates within the same family, and we wanted to be able to see if we could differentiate between family level selection or OTU or strain selection. And actually, what we saw is that for the most part they tracked together. So it looked like that selection that we saw earlier was happening at the family level rather than the isolate level, and that the isolate level is really driving more of whether you're a robust colonizer or a sporadic colonizer, not how a plant genotype is affecting you. And I think the idea of this is sort of, if you're a robust colonizer, you're really good at colonizing, and there's not a ton the immune system can do about it unless you happen to trigger something in particular, which we haven't looked at yet. Which I think is kind of, it sort of makes sense to me, I guess. But if it doesn't make sense to you, we can talk about it later. So there was one exception where this did not happen, and that was actually with the streps. So with these streps, we had 4 different isolates within the same family. 2 of them were EC enriched, so they were really good at colonizing. 1 was EC depleted, so it was not good at colonizing. And 1 was neither enriched nor depleted, so it was just sort of sporadic. It could be there or it wasn't, but it wasn't necessarily a great colonizer, so it wasn't as invasive or competitive as maybe the other things in the synthetic community. So one of the things that my lab is doing now is going back to look at these types of interactions. And so my graduate student Sarah is looking, she started looking at monoassociations and looking at plant phenotyping within this. So basically what she'll do is she'll take 6-day-old seedlings, she'll add OD of different cultures, so in this case just one thing. She'll let them grow for 7 to 10 days, and then she'll look at the plants to determine how well those isolates colonized, biomass, which is actually better in pots than plates because it's obviously much smaller on plates, and then also if the root growth phenotype changed. So this was something that she started with the streps. So here what you can see, if you'll remember back at the beginning, we saw a difference in PAD4 in Colombia with streps if we were looking at all of the OTUs. Here we're just looking at individual strains, and we didn't see a difference between them. But again, this may be something that's happening at the family level and not the strain level as a difference between the genotypes. But basically what we see is that although each of these isolates on their own can colonize about equally, one of the interesting things is the plant phenotypes they cause may be different. And so this is controlled, so this is no bacteria present. This is C-18, which was just a sporadic colonizer in a competitive community, but on its own seems to colonize fine. It looks, if you look at the roots, they look about the same as control. Although if you listen to Dave's earlier talk about image J, we could probably quantitate this a little bit better if we had been listening and not laughing so hard. And then finally, 136, which again colonizes the same as CL18, seems to be much shorter. So the plant phenotype is different, although the colonization is the same, and that's something we're sort of interested in. But something that I'm gonna show a couple slides on now is not only what are the effects on the plant, but what might the effects be on any microbes that want to get in. So streps are sort of famous for being able to make secondary metabolites. The question that we're sort of moving into is, if a strep is really good at colonizing, can it make secondary metabolites that either promote or prevent subsequent soil microbes, and there are tons of microbes in the soil, from being able to colonize. And so we've started some pilot experiments on microbe-microbe interaction, particularly with these streps. And so this was a rotation a project that I had this fall. Basically what we did is we spotted streps in the middle of plates with various dilute medias, and then we would put different test strains around them. And we did this both for soil bacteria, and we also did it with some Candida albicans strains to see if we could find some interesting antifungals in there. So when we did this, we basically saw 4 different types of phenotypes. We either saw, 1, that the growth was not different at all, 2, that we had sort of asymmetrical growth. So this one, TN19, is a good example of that. So the original spot would be the darkest part, or the most opaque part of the spot, and then you can see growth away from that. This is, we anticipate just sort of like the nutrients are taken up here, and so the nutrients are more prevalent here, and so it grows that direction. We also see an inhibitory face on some of them. So this is a strep in the middle, and this is a bacillus here. You can see that this bacillus did not grow at all in this direction, or in some cases we saw no growth at all from the spot. So this is an overview, so looking at the different medias, the streps are here, the different test strains are here. Within our test strains, we had 2 pseudomonads, which are awesome at colonizing roots. We had 2 bacillus strains, which are awesome at making biofilms, but not awesome at colonizing roots. We had E. coli, which is sort of our sporadic colonizer test that we've used several times before. And then we had a Deinococcus thermus strain, which we have never seen colonize an Arabidopsis root, and it sort of begs how we actually isolated it from a root, but that's a different story. And so basically what we're doing now is we're sort of expanding that. So for example, here with this TN29, this strep inhibiting this bacillus but not this bacillus, this is something we're now going back to test in plants. And then eventually what we'd like to do is to launch into these genomes and do some mass spec to see if we can find out what the chemicals might be that's promoting this specificity between this bacillus but not the other. And then up here with 299, the reciprocal is true. So basically, these are some studies that my lab is getting into now. And one of the cool things about making summary slides, and then that's sort of the way you have them in your mind, is that we've actually been doing everything on the right side of this. So now we can use this information that we've been generating in my lab to try to go back to the big question. So we've done 16S, but we haven't done metagenomics yet. We haven't done metatranscriptomics, we haven't done metabolomics or proteomics. And so now that we have a baseline for what's going on with these individual isolates, with the plants and with each other, we can go back and ask some, what I think are some really cool questions, but they're questions with a platform that I'm really excited about. So some future questions that we still have in the lab, going back to the salicylic acid story, is what's actually going on with the salicylic acid? I doubt it's being used as a carbon source, so figuring out first of all what form it's in the roots, as well as what it might be doing, is something that we're really interested in following up. How the plant immune system is modulating the community function, as well as just the composition, is something that we're starting to get into that I'm really excited about. And then the rest of this is focusing more on the microbe-microbe interaction. So does the plant immune system partially control the strep? So we had seen that PAD4 had different colonization, or different OTU patterns than Columbia. Is that happening when there are many streps present? It didn't look like it was happening with individual strains, but it might have just been the individual strains that we were looking at. Number 4, we're also going to look at the secondary metabolites that those streps are making to see if they might be either do they make them when they colonize the root? And then 5, what is the implication of that? So if the streps are making these secondary metabolites, is it positively or negatively influencing the colonization by other roots? And those are some experiments that Sarah is working on now. So I would now like to thank my lab at the University of Tennessee. So Jake Massey is my research technician. Sarah Stewart Sloan is my graduate student who's been working on the strep stuff. I have a variety of undergrads working on other fun little projects. And I have a former member already who's in grad school. These 2 lovely people at the top will also be going to grad school this summer. So I'm always looking for awesome people to replace them, because these are all awesome people over here. There are also awesome people on the right side of the slide. So Jeff Dangle was my postdoc advisor who took a chance on a mammalian pathologist and let her get her hands dirty and figure out what was going on in plants. We continue to collaborate on the mutant story and the function story. The same is true for Sur. He's still a graduate student in the lab. It's been really fun to sort of be able to continue conversations with them. Derek is the person who introduced me to the microbiome stuff, and for that I will be eternally grateful for him. And then this work was funded in the Dangle Lab by these people at the University of Tennessee. I have startup funds, and Susanna is a deputy director at JGI who helps us get a lot of answers with our sequencing. So with that, I'd like to thank you and take any questions.
You had to rub in the image J thing, didn't you?
I'm sorry. I couldn't help myself. It was spontaneous. I couldn't help it.
With my image J skills, I'm going to go cut that out of the video now, so I'm not going to know what you're talking about. So
that was the best trolling of a seminar host I've ever seen.
I was deciding whether to share this little tidbit, but so forever in my mind and in Mark Nishimura's mind too, I think Paul Schulte-Layford is known as pumpkin spice latte because PSL is Paul Schulte-Layford. So he will know that now.
If he watches, yeah. No, but it does happen to me every time in the fall I walk by a Starbucks. I think of him. I can't help it.
So I actually have a couple questions.
Yeah, awesome.
So, all right, these are going to be in different directions because there's a lot of data and it's kind of awesome. So, first question. So this is Arabidopsis?
This is Arabidopsis, true.
If you go to other plants, what do you think happens in other plants? Are these strains going to work the same, or is it going to be kind of completely different?
So some things will stay the same, and so I know this from another set of studies we do. We've also been working with duckweed microbiome, and so we see things that promote growth in duckweed. Some of them also promote growth in Arabidopsis, and some don't. And I think that sort of unravels some interesting questions of the things that will be shared and the things that won't. And I'll let you in on a secret that auxin is pretty universal.
Yeah, that is what I think. All right. So if you actually go out into the plant and you assume that there's some benefit, what's the minimum number of strains to actually that you find out there, and that would provide such benefits?
That's a really good question. I'm trying to think for other root microbiome studies what sort of the minimum is. I like OTU wise or isolate wise?
I just if you just go out there, what does the plant need to grow? What are Wheaties?
What's the minimum, right? I don't know, and I think the more complex it gets, the more microbe interactions you have to worry about. But then you can also have functional redundancy, which could be really cool within there. The answer is I don't know, but there's lots of things at play. Complicated, it turns out.
That's the fun of it. You have a whole career to work on that.
I hope so. Yeah.
Last two questions. So what are the ABC transporters transporting, and what is the chemotaxis receptors chemotaxing towards? So
the chemotaxic factor is got a methyl group on it. We know that, but we don't. Actually, it was discovered by my office neighbor, so I should go ask her and come back if we have time. But I don't know off the top of my head. We anticipate it's something that's the surface of the root, because the microbe it was isolated, or that was identified from, is something that chemotaxis toward roots. So we think it's something in the exudates. The ABC transporters, there's a nickel transporter, which we think could kind of be any metal. There's one for branched amino acids, one for polar amino acids, one for alternative amino acid forms. Different forms of glucose and other carbon sources. That's kind of the gist of it. We think sugar and amino acids, basically, which we know are in exudates, so that's sort of exciting.
Cool. There are no questions online. You guys, anybody else have anything?
Well, so I have a question. So you mentioned that Deinococcus strain, and that you had somehow isolated it from a root, even though it's been shown now by your data to not ever colonize it.
Yeah.
So
it could be, sorry to interrupt, I think it could be just like soil contamination. Deinococcus is known to be in rhizospheres and in soil. It's just because it can survive irradiation in like a really awesome superpower way, we tend to think it's only can be isolated from there, but it's actually also in soil.
Yeah. And I guess my last question was, I actually, way back in another lifetime, used to work on rhizosphere-competent organisms for biocontrol agents.
Yes, yes.
Right? And so with this thought, right, showing that you can have some streptomyces and sort of there's no growth impairment, are you looking for sort of tests to see that you can actually push the microbiome to go in a direction if you actually started that in a greenhouse and then put that out in the field?
Yes, we're doing that. Yeah, so those are the experiments Sarah's doing now. She's either taking a community and putting in this different streps to see if the strep can colonize. And the answer is basically the things that did well in the synthetic community can do well when there's a whole community there, not just 40 things. And the opposite experiment where she pre-colonizes with strep and then adds a whole soil community to see how that might drive it is something that she's doing. And hopefully there'll be MySeq samples soon and everything.
Excellent. Cool.
My one last comment is that somehow Lundberg and Yorstone they've gotten to look very similar to one another in your.
Yeah, they look the same in pictures, and it's really strange.
They didn't used to do that, so they
No.
had too much time together.
I imagine Lundberg is more pale because he lives in Germany now.
All right. Well, thank you very much. That was awesome.
Thank you so much. I appreciate it.
And I will splice out the image.
I mean, you could just make it its own separate video and send it to me if you want to.
David Humble's technological issues. There's some like computer.
You're just having a bad technology day. Just
Really
deal
is.
with it and don't fly anywhere.
Just not a good technology day.
And you may want to end the live feed so you don't have to trim all of this off the end too.
Oh, it's fine. This is good. This is banter. All right. Thank you very much.
Thank you. It was good to see you guys.